Hello, participant. Are you able to hear me?
Yeah.
Okay, we'll just wait for a couple of minutes. One more person is about to join.
Okay.
Meanwhile, can you introduce about yourself?
Oh, yeah, sorry. Um, hi, I'm Preeti Keeper. I'm a high school student.
So, how come you're interested in CRISPR?
Well, I've been really getting into genetics after I took biology last year, and I started doing more research on just genetics and genetic technology. So I'm really interested in learning how to use it, and hopefully I get to major in genetics when I go to college.
Okay, great. So, you're interested in pursuing a career in genetics.
Yeah.
Okay. So, you are aware of the developments in CRISPR?
What?
You are aware of the developments in CRISPR?
Yeah, I've read about like how it generally works, and like some instances it's been used, like that one woman who got cured of sickle cell anemia.
Oh, great. Maybe we'll wait for just 1 or 2 minutes.
Okay.
One more participant will join. Where are you from?
Um, the US, Connecticut.
Okay, you are from US. Okay, great. Let me share the screen. Is my screen visible?
Yes.
Okay, so let us start. It's already
Okay.
10 minutes gone. Okay, so today we will see the basics of CRISPR, right?
Okay.
CRISPR is actually a gene editing tool, right? So we will see like the basics of CRISPR, and I'll also demonstrate certain software tools. Okay.
Okay.
Okay. Now, uh, the pioneers of gene editing technology, CRISPR, are shown in this slide. So it all
Yeah.
started with Howard from DuPont. So he is credited for establishing and characterizing the CRISPR-Cas bacterial immune defense system. So
Mhm.
originally, like people were curious to know about a particular locus within certain bacterial genomes. Okay, so it was called as the CRISPR locus, clustered, regularly interspaced, short palindromic repeats. So that means it's a kind of an array of sequences where you have palindromic repeats, which are interspaced by some spacer sequences. And they could not find, like for this particular locus within the bacterial genome, they could not assign any kind of function initially before 2009. That's what for years they've been thinking that it's a kind of a junk DNA. But bacterial genome being quite small, it does not have place for junk DNA, right? For example, if we take the human genome, nearly 98% of the human genome is non-coding, right? And only less than 2% is coding sequence. So, but in the bacterial genome being very small, it is not the case. So when you have a loci within the bacterial genome, which is not associated with any function, people thought initially it was junk, but later people thought like, bacterial genome is already like, if you see for multiple genes, bacteria has one promoter, correct, na? Right? So the thing is like, why you should have some loci which is not functional? So they started trying to analyze and reason out, and then they did some bioinformatic analysis, and then they found that these CRISPR loci, they actually have spacer sequences uh, uh, between those repeats, and these spacer sequences have more in sequence similarity with the bacteriophage genomes. Okay. So then this led to the theory or speculation that, like maybe these viral bacteriophage sequences have been acquired during some infection, and that they have been integrated into the CRISPR loci. Then they started dwelling like how this can be uh, useful for the bacteria. Then they found that, like this acquisition of bacteriophage sequences as spacers into the CRISPR loci in between those palindromic repeats actually gives rise to a quite a powerful immune defense system against the invading nucleic acids. Okay, so nucleic acid based infections like bacteriophage infections. That means whenever a bacteriophage tries to uh, you know tries to invade a bacteria, so it will try to push in the nucleic acid inside. It will pump the nucleic acid inside. Okay, so by sitting in the cell wall, it will try to insert the nucleic acid inside, and the nucleic acid, when it's coming inside, it will try to either cause a lytic cycle in the bacteria, or it will cause a lysogenic cycle, where integration of the phage genome happens into the bacterial genome, some part of the phage genome. So the thing is uh, the Howard researchers, they found out Howard from DuPont, he established the fact that this CRISPR-Cas system actually constitutes a important immune defense system. It's an adaptive immune defense system, particularly to safeguard against the bacteriophage infections. Okay. So later, in 2012, Jennifer Doudna and Emmanuel Carpenter, they actually published a paper in Science. So they actually established the fact that this CRISPR system can be repurposed uh, for important applications. So it can be repurposed into a gene editing tool. So what is actually a immune defense system in bacteria? Not in all bacteria, in certain bacteria, like 30% of bacteria and 50% of archaea are known to contain these CRISPR systems. Okay. And so Jennifer Doudna and Carpenter, they showed the fact that these whatever is there in the CRISPR as a battle defense system can be repurposed and reprogrammed into a gene editing tool, and which can be used in all types of cells. Okay, so this is actually a significant achievement, and therefore they have been awarded the Nobel Prize for that. Okay, so they established the fact that this battle defense system, the CRISPR, could be turned into a cut and paste tool for editing gene sequences. Okay, so they established the fact that by just changing some region of the guide RNA. Okay, so by changing the sequence of the guide RNA, one can target a gene, any gene of interest. Okay, so now this bacterial defense system, what is used, what evolved in bacteria, now has been repurposed and reprogrammed, and it is now more applicable in editing mammalian cells and other cells. Okay, in fact, the application of CRISPR in editing mammalian cells and human cells is more, compared to editing bacteria with the CRISPR. Okay, so like CRISPR can be used to edit other bacteria also, and even yeast, and like plants, and also human and mammalian cells. Okay, but the application in gene therapy is actually a very important application of CRISPR. So CRISPR is also used in diagnostics, right? CRISPR based diagnostic kits are available. You might have heard about the COVID kit, right? COVID kit. So these scientists also set up companies which manufacture those kits, diagnostic kits, and also they are into the gene therapy program for several diseases. Okay, so particularly Zhang from MIT, he actually was one of the early pioneers to use CRISPR as a tool for developing diagnostic kits against viral diseases, and also to come with a gene therapy program to treat genetic diseases. Okay, particularly CRISPR is very suitable for mononuclear genetic diseases, where a single gene is responsible for the mutation in a single gene is responsible for the disease. For example, you told about the sickle cell disease, right? This is a very good example, right? Hello?
Yeah. Yeah.
Yeah, you know how this sickle cell disease happens?
I think so. It's like some sort of mutation when red blood cells aren't made properly.
Mutation where?
What?
No, you
Oh,
told about
um,
mutation, right?
yeah, like red
Mutation
blood
where?
cells. Yeah.
Where the mutation occurs?
What I said, so like when red blood cells like aren't made properly, so they have like that sickle shape.
These are the manifestations of the mutation. But where does the mutation arise? So the thing is, any genetic disease will have some mutation, mutation in a gene, which
Yeah.
impairs its function, right? So if you see in sickle cell disease, you will be having this hemoglobin beta gene, right? Hemoglobin is a protein which has this oxygen transport function, right? And
Yeah.
hemoglobin itself is a tetramer. It has two alpha chains and two beta chains. So the alpha chain is coded by the hemoglobin A gene. Okay, hemoglobin A gene. And the beta chain is coded by the hemoglobin B gene. Okay, so two chains of hemoglobin B chain and two of the alpha chain, alpha and beta chains. Okay, so they combine to form a tetramer. Okay, and suppose what happens is in the beta chain. Okay, in the beta chain, in the sixth amino acid, it should be actually glutamic acid. The sixth amino acid in the hemoglobin B chain should be glutamic acid. You know the properties of glutamic acid?
No.
Glutamic acid is an amino acid which actually has a negative charge. Okay, so it's a negatively charged amino acid, and it sits in the surface of the proteins. It's not buried inside the core of the protein. Okay, so this glutamic acid is actually mutated to valine. Okay, so this valine is a hydrophobic amino acid, and generally it is not present in the surface of proteins. It's generally buried in the core of the protein. Okay, being hydrophobic and branched amino acid. So this mutation is actually happens because of a single nucleotide change, an SNP, single nucleotide polymorphism. So if you see the genetic code, just one nucleotide change, brings about this disease. Okay, so one single nucleotide change actually changes the glutamic acid to valine. Okay, have you seen the genetic code? Do you know about the
Um,
genetic code?
I think like the
Huh?
DNA RNA protein thing.
Yeah, the genetic code. Genetic
I
code means like in the mRNA, the codons will be there, and what
oh
codon will code for what amino acid.
oh yeah, I've seen that.
Okay, just I'll show the genetic code. Are you able to see my screen?
Yeah.
Just 1 second. I'll just show the PPT. I think you're seeing the PPT, right?
Yeah, I'm seeing the people.
I'll just show the screen. Are you able to see this one?
Yes, I think it's loading.
Yeah. So genetic code actually, like in the mRNA, the sequence will be there, and that mRNA will code for a protein. So three nucleotides at a stretch will be taken, and the ribosomes will actually put a particular amino acid in the growing protein chain. That means like the sequence of the mRNA is read as triplets, three nucleotides in a stretch, and for each three nucleotide, a corresponding amino acid is added. So this relation between the codon and the protein and the amino acids is given here. That means whenever you have these codons in the mRNA, these kind of amino acids are formed. For example,
Mm.
if we take, I was telling you about glutamic acid, na? Right?
Yeah.
So in the sixth amino acid in the sixth amino acid in the sixth amino acid in the beta chain, okay? Sixth amino acid in the beta chain will be GAA, which codes for glutamic acid. So originally, the sixth amino acid in the beta chain of hemoglobin is GAA. Okay. So I'm talking about the gene, the beta gene. Okay. So it will be GAA. Now this mutation happens. So GAA becomes GTA. Okay. A to T transition happens. Single nucleotide change. And because of that, like in the mRNA, GUA is formed. When it is GTA in the DNA, GUA will be the mRNA, right? Yes or no? Your
Yeah.
answer. So that will code for valine. Okay?
Mm.
That will code for valine. So what happens is glutamic acid codes for glutamic acid is mutated to valine. Okay? Glutamic
Mm.
acid is mutated to valine. So this is what is happening actually. Glutamic acid is mutated to valine. So now, this is actually a mononuclear genetic disease because a single gene is attributed uh, as responsible for the disease, right? The hemoglobin B gene. Okay. So CRISPR is actually well suited for this because, we can actually try to edit this, like whatever's wrong mutation is there, we can revert it and we can cure the disease. Okay. So the advantage is being a mononuclear disease, and that too, we can take the hematopoietic stem cells from the bone marrow of the patients, and then you can do CRISPR based editing outside the body. Okay. So you can take the hematopoietic stem cells from the bone marrow by apheresis, and then you can actually do the CRISPR editing in the lab. And then once these cells, are edited and this mutation is reversed, okay, so then actually the edited cells can be again put back inside the patient, and then they will have a complete cure. Okay, so patients who've been treated this way by the CRISPR-based gene therapy, they have shown complete cure, and almost like for the last 3-4 years, they're leading a normal life. Okay, so this is the advantage of CRISPR-based gene therapy. Okay, going back to our slides. Yeah, so you're able to see, right? So the Nobel Prize in Chemistry was awarded to these scientists because they were the ones who established the fact that one can repurpose and reprogram this guide RNA, and you can literally edit any gene that you want, okay, in any organism. So that is why they got the Nobel Prize for that. So So basically, what is CRISPR? So in CRISPR is actually a clustered, if you can see in the bacterial chromosome. Okay, as I told, nearly 30% of the bacteria and
Mm-hmm.
nearly like some 50% of archaebacteria, which are called extremophiles, like halophiles, acidophiles, okay, thermophiles. So those kind of archaebacteria, like 50 to 60% of archaebacteria, they actually contain this CRISPR locus. CRISPR locus is nothing but a region within the bacterial chromosome which contains this kind of array, CRISPR array. Okay, so as I told initially, they were puzzled why this CRISPR array is there and what is its function. And then they established the fact that this actually plays an important role in the bacterial immune defense system to safeguard those bacteria against the bacteriophages. Okay, because you have already restriction enzymes and all those things in the bacteria, but they are not very effective against phage infections. Okay, so they are not very specific also. So that is the reason, like some bacteria, they have evolved this CRISPR system. Okay, so if you take this CRISPR loci, okay, you will see that it contains all these elements. It will contain the Cas genes. Okay, the Cas genes code for the Cas proteins like Cas9, Cas12, Cas13, Cas1, Cas2. These are these are Cas proteins. Okay, CRISPR associated proteins. And then you will have a CRISPR array, which array in the sense like some kind of repetitions are there. So we will see repeat sequences, palindromic repeat sequences. You know about palindromic, right? Palindromic.
I think so. It's like they're the same forwards and backwards.
Yes. So yeah. So so the thing is like in the case of DNA, you read from this direction and you read from this direction. It's going to be the same.
Yeah.
Okay. So for example, if it's G here and it's G here, if it's C here, okay. So if it's
Mm-hmm.
C here and if it's T here, it's also T here, like that. Okay.
Yeah.
And the other one will be here A. So this will be A, this will be G, this will be C, right? And what will be here? This will be A, this will be G, C. So it's a double stranded DNA, na?
Mm-hmm.
Like two strands of the DNA, right? Two strands of
Mm-hmm.
the DNA. If you see this way also is GCTAGC. This way also is GCTAGC, right? So in a double stranded DNA, you have this kind of thing. It's called a palindromic. Okay, so these palindromic sequences are there, and these are repeat sequences. That means like this same sequence is repeated multiple times here, 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 here. Okay, so you have this palindromic repeat sequences, and these repeat sequences are around 20 to 40 base pairs. Okay, 20 to 40 base pairs. Palindromic sequences. Okay, and palindromic sequence 20 to 40 base pairs. And then you can see between those repeat sequences, you have spacer sequences, like which separate the one between those repeat sequences are called spacers. Okay, and the spacer sequences can be from 20 to 15 base pairs. 20 to 15 base pairs. Okay, so the CRISPR repeat sequences are 20 to 40 base pairs, and the spacer sequences between those repeats, they will be uh, 20 to 58 base pairs. And now these spacer sequences are all unique. That means the spacer here will be different from the spacer here, the sequence. Okay. And these are all spacers now. They're all different in sequence. Like every spacer is unique. Okay. But the repeat sequences are the same actually. The same sequences are repeated. Okay. Those shown in as the gray color boxes. Okay. So those are the repeats. And before this CRISPR array, you have a leader-like sequence, leader sequence. This leader sequence actually acts like the promoter sequence, okay, where which regulates the expression of the CRISPR RNA. Okay, so it acts like a like a promoter sequence. So this is actually the CRISPR locus. So CRISPR is nothing but clustered. It's all clustered in one place, right? So clustered, regularly interspaced. They are interspaced, right? Regularly interspaced short palindromic repeats. So you have palindromic repeats which are interspaced, okay? And they're all clustered in a part of the bacterial chromosome. So that's why clustered, regularly interspaced, short palindromic repeats. Okay, is it clear? Hello, participant, are you there?
Oh, sorry, I thought I had unmuted. Yeah, I get it.
Okay, so now people, what they did. So this is another illustration. Okay, so the question is how this spacer got acquired here. When they did a bioinformatic analysis, they found these spacer sequences. Okay, these spacer sequences, they are quite similar to the phage sequences, bacteriophage sequences. Okay, so
Sorry,
if you take
wait,
the back,
the what sequences?
huh?
You said they're similar to the what sequences?
The spacer sequences, the CRISPR
Oh,
spacers, no? Between
yeah.
those repeats, you have spacer sequences, right? Those
Hmm.
spacer sequences, the sequence actually is having high similarity with the bacteriophage sequences.
Oh, okay, okay.
Okay, so this led to the speculation that maybe during the bacteriophage infection, some portion of the bacteriophage sequence would have been integrated between those repeat sequences. Okay, and that became the spacer. So every time that bacteria is subjected to a new infection by a phage, okay, bacteriophage. So some small portion of that phage DNA is getting integrated, and then this becomes the CRISPR array. Okay, so this was a speculation. So you can see like phage comes and it invades the bacteria. So it inserts the nucleic acid inside. Okay, so a small portion of the nucleic acid is cut. There are some special mechanisms which are still yet to be unraveled, which actually ensure that a small portion of the phage genome is cut, and then it gets integrated into the uh, CRISPR array as a spacer. Okay, so this is what you see in this red color here is actually a newly acquired spacer. Okay, and the ones which are shown in yellow here, yellow spacers, they are actually phage sequences which have been acquired during the previous infection. Okay, so that means like this immune system. I will tell why it's an immune system in a short while. But this supposed immune system is adaptive. That means every time a phage infection happens, it can adapt and it can develop immunity against that phage. Okay, so it's a kind of adaptive immune system. So certain bacteria like you know, have you heard about bacteria like Streptococcus pyogenes? Okay, Streptococcus pyogenes has a very well developed CRISPR system. Okay, so it has around 120 spacers. How many? Nearly 120 spacers have been found. 120 spacers. Okay, so 120 space that means like it has immunity against several types of phages, bacteriophages. Okay, because like if it has 120 spacers and all these spacers are all unique, right? Like in the sense like each spacer has a unique sequence. That means like they have been acquired by during several phage infections. So that means like that SP Streptococcus pyogenes has immunity against several bacteriophage infections. Okay, so this is actually the mechanism of the acquisition. Okay, now in a more detailed way, you can see this natural CRISPR pathway. Okay, so you can see the bacteriophage. Okay, that sits on the bacterial cell wall, and through the bacterial cell wall have porins. Through that, actually, it will inject the DNA, its nucleic acids. So the nucleic acid, the DNA, which is called the invading DNA, that actually is cut. Okay, so DNA fragments from the invading DNA are incorporated into the CRISPR locus as spacers. The exact mechanism of incorporation still remains unknown. Okay, so though what actually causes this cleavage of invading DNA and how it is getting integrated, those studies are still going on. Okay, but the application part of CRISPR has actually become more got more attention and more funding, and therefore people actually now they're concentrating in the application part of CRISPR rather than how the CRISPR evolved in bacteria. Okay, so by some mechanism, like the invading DNA is cut and then it got incorporated into the CRISPR locus, the spacer. Now, you can see here the black ones in this picture, they represent the repeat sequences, and the gray one, the spacers. The red one, the newly acquired spacer. Okay, and so this is the CRISPR locus. Now, this CRISPR locus will, you have RNA polymerase. Now, they will actually come and, scan this one, and then they will make the RNA, right? So, this CRISPR locus will code for the pre-CRISPR RNA. The pre-CRISPR RNA, as you see, like it's a single stranded long RNA molecule. So, the entire array is made into a pre-CRISPR RNA. And now, you saw now that before the CRISPR array, you have the CRISPR-Cas genes and all. Na? So you have a track RNA formed from there. So track RNA is nothing but transactivating CRISPR RNA. This is another RNA molecule formed by the expression of some Cas gene. Okay. So the track RNA, you can see the track RNA has a stem loop-like structure. Okay. And this track RNA, which is called as a transactivating CRISPR RNA, it also contains a anti-repeat sequence. So this sequence will be actually complementary to the repeat sequences found in the CRISPR locus. Okay. So when the pre-CRISPR RNA is formed, the repeat sequences within the pre-CRISPR RNA, they can actually, for example, whatever is shown in black now, they are the repeat sequences. So they can actually bind to this anti-repeat sequence of the track RNA. So that means a complementary base pairing between the repeat sequence of the pre-CRISPR RNA and the transactivating RNA can happen. Okay. So what happens is at the same time, there are some nucleases which act on this pre-CRISPR RNA, particularly this RNase 3, CSN1, all these things. They will come and act on the pre-CRISPR RNA, and they will cut in such a way that you will end up having at least one spacer and one repeat. So they'll basically they will cleave like this. So you'll end up having one spacer, one repeat. Okay, one spacer, one repeat. So what happens is now this track RNA having a region complementary to the repeat, it will start annealing to that, right? And then this duplex of the two RNA molecules, the CRISPR RNA, which is got from the pre-CRISPR RNA, and the track RNA, okay? So when these two form a duplex, you will get the guide RNA. You're understanding?
Yeah.
Is it clear?
Yeah.
So in the naturally, in bacteria, whatever you find in the CRISPR, so it will have a guide RNA, which is actually a duplex. Duplex means made up of two RNA molecules. One is the CRISPR RNA. The CRISPR RNA itself has two elements. Like it's a single chain, but it will have two elements. One is the spacer sequence and one is the repeat. And to the repeat sequence of the CRISPR RNA, you will have the transactivating CRISPR RNA, which is bound to it, okay, binding to it. So these form a duplex, and the duplex is called as a guide RNA. Now you can see like whatever shown in green, the track RNA, green color. So that actually has a binding affinity to the Cas9 protein. In the Cas9 protein is a multi-domain protein. It has several domains. Okay, each has a different function. Now, this this guide RNA, okay, so this guide RNA is like, for example, the trans-activating CRISPR RNA, it is able to bind here. Okay, it's able to attach to this Cas9 complex. So this duplex guide RNA, it's actually, for example, I can say this entire thing, the repeat with the trans-activating CRISPR RNA. This entire thing comes and binds to the protein, the Cas9 protein. Okay, the spacer sequence will be actually free outside. So and therefore it's now available for binding to the DNA. Okay, whereas this portion, the repeat and the track RNA, these actually constitute the scaffold. We call it a scaffold sequence, because this region will be like will be the same for every guide RNA. Okay. The only thing is this will change because the spaces are all from are having unique sequences. Now they're all having their own sequences. So spacer sequence will keep on changing, but the repeat and the track RNA will remain the same for a given Cas9. Okay, so this region we call a scaffold because this is the one which gets bound to the protein. So when this guide RNA binds to the Cas9 complex, Cas9, this actually uh, undergoes a conformational change. So there's a domain rearrangement happening. You can see the domains which are shown in different colors, right? We can see the relative reorientation after the binding event. This has gone here, this has gone here, right? This has gone here, and you can see this yellow color one has come here. So domain rearrangement happens, and now it is actually called as the guide RNA complex or ribonucleoprotein. The RNA, guide RNA bound to the protein, ribonucleoprotein. So it's called as the active conformation of the Cas9. Okay. So now the spacer sequence, which will be there, the spacer sequence, that is actually not bound to the protein. Okay. So that is free to, that is free to actually anneal with the DNA. Now this protein, right? The protein with the guide RNA complex will actually be able to bind to the DNA. Okay, first the protein binds to the DNA. So this protein has, can you see this orange color domain here, right? So this is called as a PAM interacting domain, and this will identify some unique stretch of sequence within the DNA, target DNA. For example, if it's SP-Cas9, Streptococcus pyogenes Cas9, it will identify NGG sequences within the invading DNA. Okay. So this NGG sequence can be at multiple places. So it will go and bind to those places, and then it will cause local denaturation, so that the spacer sequence of your guide RNA will be able to anneal with one of the strands of the DNA. Okay. So once this annealing of the guide RNAs, so this region, the spacer region, we call it the target complementary region of the guide RNA. So the target complementary region of the guide RNA, once it's able to anneal with the uh, with what? Anneal with one of the strands of the target DNA. Okay. So the target DNA strand which anneals, so we call that region as a protospacer region. Okay. So because the spacer region is binding with the protospacer region of the target DNA. So once this annealing is perfect, that means all the 20 bases of your target complementary region of the guide RNA is established base pairing with the protospacer sequence of the target. Then what will happen is, again, this Cas9 will undergo a conformational trigger, leading to the activation of the nucleus domains. So the nucleus domains, you have a pair of nucleus domains. The one shown in green color here and yellow color here, they are the nucleus domains. And these two nucleus domains, they can cleave the two strands of DNA. Okay, almost at the identical position. So what happens is, then you get, cleavage of the DNA. Okay, are you comfortable with this?
Yeah.
Understood, right? So this is actually the natural CRISPR pathway which is happening in the bacteria. The phage comes, the phage will inject the nucleic acid. Then some unknown mechanism is happens to take a small fragment of this invading DNA and integrate into the CRISPR locus as a spacer. Now the CRISPR locus will undergo transcription to form the pre-CRISPR RNA. The pre-CRISPR RNA will actually undergo cleavage by some enzymes like RNase 3, CSN1, leading to shorter fragments which will have at least one spacer and one repeat. These shorter fragments are called CRISPR RNA. The CRISPR RNA will form a duplex with the another RNA molecule called as the trans-activating CRISPR RNA. So the repeat sequence of the CRISPR RNA is actually the one which anneals with the track RNA. Now this is called as the guide RNA now. Okay, the complex between the CRISPR RNA and the track RNA is called guide RNA. Now this guide RNA will be able to bind to the Cas9 protein. The Cas9 protein is itself is a produced from the Cas genes which are on the CRISPR locus. Now this triggers a conformational change. Now the Cas9 becomes an active conformer. Now the Cas9 along with the guide RNA, it will go and bind to the DNA at some certain sites. Okay. So that will depend on the type of Cas9. So if it's SP-Cas9, Streptococcus pyogenes Cas9, it will identify NGG. N could be any nucleotide, GG or guanines, right? So so it will identify NGG and bind there. And after binding, what it will do? It will try to denature the denature means the double stranded DNA will be made to single strand. Okay, just a few, few like this 20, 30 base pairs before that PAM sequence will get denatured because of this protein, Cas9 protein, and then the guide RNA, the target complementary region of the guide RNA, or we call the spacer sequence of the guide RNA, that will be able to anneal with what? That will be able to anneal with
What? I don't.
that will be able to anneal with the target DNA, right? So a single strand of the target DNA, and once this annealing is ensured, then the nucleus domains they get activated, and they will cleave the both the strands of the DNA at the same place. Okay, so you'll get this kind of blunt and cut. So this cleavage happens 3 nucleotides before the PAM sequence. To the PAM sequence only the protein comes and binds. This orange colored domain of the protein comes and binds, recognizes this PAM sequence and binds there. Then it's locally these sequences are denatured. Then after denaturation, the guide RNA target complementary region will form base pairing, and then it will actually cleave 3 nucleotides before the PAM sequence. Okay, and this will lead to cleavage of DNA. So this is
Mm.
the one which is happening in bacteria. Okay, now people have uh, now repurposed this. Okay, so if you see, this is the natural CRISPR pathway that I explained today. Now, people, as I told, right, Jennifer Doudna and Carpentier, their work involved taking this natural CRISPR pathway and then trying to establish it as a gene editing, general purpose gene editing tool, which can be used to edit mammalian cells or plant cells or yeast cells, whatever. Okay, so how this engineered CRISPR-Cas9 system, which find all the applications, right? So this is actually possible because we try to create a plasmid which will code for the Cas gene, the which will code for the Cas protein. So the Cas9 gene is there inside the plasmid under a particular promoter, and similarly the guide RNA, okay, that will be also under another promoter. For example, a U6 promoter. So, for example, I want to edit cells. I will use a U6 promoter for expressing the guide RNA and a CMV promoter for expressing the Cas9. Okay, and this can be sent inside a human cell, human cell, and then that will do the editing. How? because this plasmid will code for the guide RNA as well as the Cas9, and these will form the complex inside the cell. In situ complex formation will happen of the guide RNA and the Cas9, and that will lead to the active Cas9. So the Cas9 will go inside the nucleus, right? Because the Cas9 will be having a nucleotide localization signal, nuclear localization signal. So it will help the entire Cas9 protein to go inside the nucleus. So using its guide RNA, so it will bind to the particular PAM sequence in the DNA, and then denaturation occurs. Then using the target complement region of the guide RNA, it will be able to anneal to the protospacer, and then it will cleave. Okay. So here, for the sake of convenience, what they did is the two strands of the guide RNA, right? So this CRISPR RNA and the track RNA, they have added some few bases and they have joined it like this. Okay. So this is what is there in the engineered forms, man-made forms of guide RNA. We, instead of expressing as two single pieces, two separate pieces of guide, two separate pieces for the guide RNA, now we actually make a single piece of RNA. Can you see this one? It becomes one single
Yeah.
piece just by adding some nuclear and fusing them. So because when it becomes single piece, the activity remains the same. The activity remains the same, but the advantage is we don't need to, for example, if you want to express these RNA molecules, I need to have two promoters, one for expressing the CRISPR RNA and one for expressing the track RNA, and then they will form. They will bind and form a complex. So the thing is like to simplify things, they have just added some few bases here. Okay, and this is now called as a single guide RNA. So single guide RNA is not found in the natural system. Single guide RNA is an engineered system. Okay, so we just for our convenience we add some few nucleotides and make this as a single strand of RNA. So it's called as a single guide RNA. So the single guide RNA can be expressed under a suitable promoter. For example, this U6 promoter can express the entire guide RNA. Okay, so they clone it downstream of the U6 promoter, the DNA sequence of the guide RNA here, right? So this spacer sequence can be seen here, and this remaining guide sequence can be seen here. Okay, so this single guide RNA is found in the engineered systems, the one we try to design. Okay, so this will bind the same way as the normal guide RNA. So this will go and bind to the inactive Cas9 complex, and then a conformational change will happen. It'll become the active form. Okay, so this is what is happening in the engineered systems that we try to develop. So Jennifer Doudna and they were the first to develop these kind of systems and show that by changing this spacer sequence. Suppose I change this spacer sequence, then I can edit some other gene. So depending on what gene I want to target, I just need to change this whatever shown in red color, this sequence. Whereas the green sequence will remain the same. I need not change the green sequence. Okay, so the scaffold sequence will not change, but the whatever shown in red, that will change. The sequence will change depending on the gene I want to target. Okay, so I will identify some unique region within a particular gene, and that sequence will be grafted here. And now this guide RNA, which is formed by that alteration, that will actually end up binding with the Cas9, and then recruiting the Cas9 to go to the particular protospacer there, and then cleave that one. Okay, so this is how it functions. Any doubts here?
No.
Okay, to tell in detail. Okay, so yeah, so this is how it binds, right? So you have this Cas9 complex. This Cas9 complex, if you see, it has multiple domains. Can you see the structure? It has a receptor 1 domain. It has multiple two nucleus domains, H and H and rho C. H and H is a nucleus which will actually cleave what? It will cleave
Uh,
the DNA strand which is complementary to your guide RNA. If your
Mm.
guide RNA is able to form a complementary base pairing with that protospacer sequence, then that strand which forms complementary base pairing with the guide RNA, that will be cleaved by the HNH nuclease. And the RuC nuclease will cleave the other strand of the protospacer. Okay. And so these two are nucleus domains. The receptor 2 domain just facilitates the binding of guide RNA to the receptor 1 domain. It has no function on its own. And the PAM interacting domain, it actually is the one which binds to the DNA. So this is the DNA binding region of the protein. And the receptor 1 domain is the one which binds the guide RNA. Right? The scaffold sequence of the guide RNA is bound to the receptor 1 domain of the protein. And this bridge helix, each actually relays the conformational change. Okay, so it uh, helps to activate the nucleus domains once the uh, you know once the guide RNA is successfully annealed with the target DNA. Okay, so it acts as a conformational relay to to activate the nucleus domains for cleavage. Okay, so that means the activity is highly controlled. Cas9 alone cannot cleave. Okay, so Cas9 along with guide RNA, okay, just binding to a DNA will not cleave, though it has a guide RNA. So Cas9 with the guide RNA bound to it has to bind to a DNA, and even that will not ensure a cleavage. So after binding to the DNA, it should actually denature the sequence, and then the guide RNA's spacer sequence should actually form complementary base pairing. The 20 bases of your guide RNA spacer sequence should actually anneal with the 20 base pairs of the protospacer. If this annealing is perfect, then only the activation of HNH RuC nuclease happens, and these will start cleaving. Okay,
Mm-hmm.
so that means like it's a kind of a very controlled way, controlled mechanism of cleavage. It will not cleave just like that. Okay,
Yeah.
so so if you see here, people like if you go to PDB, PDB, right? Protein Data Bank.
No.
Okay, so you have something called protein data bank. Okay, so for example, we use this RCSB, Royal Chemical Society of Britain Protein Data Bank. It contains more than 2 lakh protein structures whose structure has been solved by experimental methods. Okay, so you have very high resolution three-dimensional structure of proteins which are deposited in the protein data bank. That means every atom in the protein, their coordinate position is known. Okay, so how the 3D structure is known. So now this 3D structure of the Cas9 protein alone, the 3D structure of the Cas9 protein bound with the guide RNA, and the 3D structure of the Cas9 protein bound with the guide RNA, which is bound to the target DNA. All three structures are available in the PDB. Okay, you can go. Can you see this? 4-CMP, 4-ZTO, and many more are there. Okay, so you can just go and compare those structures. You will see that there's a these are experimentally derived structures. Okay, by crystallography. So you will see that there's a conformational change which is very obvious, going from the Cas9 to the Cas9 which is bound to the guide RNA to the Cas9 guide RNA complex which is bound to the target DNA. So there's a conformational change in all these events. Okay, so that is what is actually uh, experimentally proven. Okay, so to illustrate the mechanism of action. Now, if you take this target DNA, right? So this is the target DNA. What you can see, this is the target DNA. Can you see? This is the target DNA. This is the target DNA. And where is the PAM? PAM can be anywhere, like multiple places. So the PAM for this SP-Cas9 is the NGG, right? N could be any nucleotide. So this is also a PAM, right? TGG is a PAM, correct? Na? This TGG could be a PAM.
Yeah.
Now, so suppose the Cas9 binds to this TGG. So first it clings to the TGG. Then what will happen? It will try to denature. You know what is denaturing, right? It will try to
Yeah.
break the hydrogen bonds, and the double-stranded DNA will become single-stranded, at least locally. Locally, it will become single-stranded. So that means like a few base pairs, they will get distorted. The hydrogen bonds gets broken. Okay, this is because of the pulling mechanism of the Cas9 protein. Cas9 protein will actually has some loops, phosphate lock loops, and they will try to pull and unzip. That means like the like unzipping process. Like it will try to denature the double strand into single strands. Now what will happen is your guide RNA, which is bound to the Cas9 protein. So this guide RNA is shown here. This entire thing is the guide RNA. Entire thing is the guide RNA, the blue and the red one. The blue one is the spacer sequence of your guide RNA here. Okay. And so in this picture, the blue sequence is the spacer sequence. So what happens, you can clearly see. So this is actually annealing. Once this is denatured to one strand, okay, generally the strand which is actually opposite to the PAM sequence. Okay. So there, this guide RNA's target sequence will anneal. The target complement region of the guide RNA will anneal with this sequence. Okay. Once it's annealed, you can see the red region. What is the red region? This is scaffold, right? The scaffold part of the guide RNA. So guide RNA, this is a spacer sequence of the guide RNA, and this is a scaffold region of the guide RNA. Okay. So scaffold region is bound to the protein, and this spacer sequence is able to anneal with the protospacer sequence, one strand of the protospacer. Now, when the annealing is perfect, all the 20 bases are matched. Okay. Then the nucleus domains cleave, and they will cleave 3 nucleotides before the PAM sequence. So this is the PAM. So it will cleave here and here. So that will lead to a double-stranded cleavage. So this is the schematic picture. You're able to understand?
Yeah.
Any doubts?
Um, it's like what exactly is a PAM sequence?
Yeah, the PAM sequence, as I told, for this Cas9 protein to bind to the DNA, it recognizes some patches. Okay, it cannot bind to any DNA sequence. So if the Cas9, for example, if it's SP-Cas9, Streptococcus pyogenes Cas9, it will look for some unique sequences, like for example NGG. That is the sequence which it identifies, and it'll go and cling there. Okay, but so these are all potential PAM sites. Okay, but actually, so there may be multiple places you can see PAM sequences, right? Because the possibility of seeing NGG two guanines together, it's quite a high, right? So every 10 base pairs, every 10 to 12 base pairs in the DNA, you can see a NGG. Now the question is, it can potentially bind to all these places, but upon denaturation of the sequences before it, and then followed by the guide RNA's target complement region to anneal with one of the strands, and only if that is successful, then the cleavage will happen. Otherwise, it will move on to the next PAM, right? That means in the DNA, in the target DNA sequence, you'll have multiple PAM sequences. NGG could be in multiple places, right? For example, this is also NGG. Can you see?
Yeah.
N could be any nucleotide. This is also NGG. It also can bind here also. But the sequence ahead of it, if it denatures and then tries to anneal, it will not anneal, right? Because your guide RNA is not matched to that one. So it will start moving to the next PAM. So it can actually literally scan the target sequence. And so since this is the spacer, this will be the target sequence where it binds, that will be the protospacer. Because based on that only, we have designed the guide RNA sequence, right? And the one which is adjacent to it, okay, which is used for recognition, that is called the protospacer adjacent motif. So this is the target sequence is the protospacer, right? Protospacer, proto. So this is the protospacer adjacent motif, which is used for recognition by the Cas9 protein. Okay, and this based on this protospacer sequence only, we have designed the guide RNA's target complement region. That means I identify a gene. Okay, this is the gene I want to knock out. Then I take the sequence of gene. I identify some unique region, a 20 base pair stretch within the gene, which is unique to the gene. And then that sequence is taken and grafted in as the target complement region of the guide RNA. The remaining part of the guide RNA will remain the same. The scaffold region will remain the same. But the first 20 bases from the 5' end, it will be, it will depend on what you want to target. Okay. So once that is grafted, then, like that will go to the particular target DNA. And then using the PAM site, the protein will bind there. Then denaturation happens. Then annealing will happen with the guide RNA. Then the cleavage will happen. Okay. So this is what's happening. Now, uh, after the cleavage, so cleavage has happened. Now, this is what Cas9 can do to cleave, right? But after that, what is happening? Any idea?
Um, no.
Yeah. So to summarize, you can see this picture. Like, can you see here?
Yeah.
Is another view of the same process. So you can see that, like you have an engineered system where you have a Cas9 and a guide RNA under a promoter, under a plasmid in a plasmid. This plasmid is, transfected into the
Yeah.
mammalian cell. And then this will code for the Cas9 and the guide RNA, and they will form a complex. And this complex will actually come and it will bind to the NGG sequence, the PAM sequence. Okay. First, the Cas9 will cling to the PAM sequence. Then it will locally denature the strands, double strand DNA. So this denaturation happens, then this allows the guide RNA. So the guide RNA's first 20 base pairs, okay? So what we call as the target complementary region or the spacer region of the guide RNA, that will be able to anneal to one of the strands. If the annealing is perfect, okay? If the annealing is perfect, then the cleavage will happen. And the
Okay.
cleavage will happen 3 nucleotides before the PAM sequence in both the strands. Okay, so this is the mechanism of action of the conventional Cas9. But there are many Cas9s which have been discovered, and they actually can cleave sometimes slightly differently. For example, a different PAM sequence. For example, I was telling about SP-Cas9. But if we go for some kind of Cas9s like a F1 Cas9 or CPF1 type of Cas9s, they actually identify a different PAM sequence. Okay, it's not NGG always. Okay, so the other so the Cas9s coming from other types of bacteria, they may recognize other different PAM sequences. Okay, other than NGG. And also the cleavage site will vary. Instead of cleaving 3 base pairs before the PAM sequence, they may cleave elsewhere. Okay. For example, in the F1 CPF1 kind of Cas9s, the cleavage happens outside the target region. So this is the target region where the guide RNA binds. This is the PAM. Okay. But the cleavage happens somewhere here. It's not in the region where the guide RNA binds. Okay. So so we call it PAM in, PAM out. So PAM in sequence, PAM out sequences. So so certain Cas9s cleave out of that region. Okay. So there are always like several Cas9s have been discovered now. Okay. So what I'm explaining is the most conventional, well-studied Cas9s. Okay.
Yeah.
Any doubts?
I don't think so.
Yeah. Now we can go to the next thing. Now, okay, cleavage is happening. So cleavage has happened. A double stranded cleavage has happened. Okay. Now what next? So if you take a human cell, okay, suppose you cultured some human cells, so and you're transfected with the Cas9 complex, then that has successfully cleaved. Now this cleavage has to be repaired, nah? Right? Whenever there's a double stranded DNA break, the cell will try to repair that. Okay. So the Cas9 has done its part. It came and it identified, it cleaved. Now the cell has to repair this cleavage because a double stranded cleavage can prove costly for the cell because a double stranded cleavage will expose the ends. So the exonuclease will start chewing from those ends. Okay. So that is why we need to, the cell needs to act quickly and then try to fix this double stranded break. So it does some kind of quick fix. Okay, we can say a dirty quick fix, and it actually recruits some proteins which will bind to either ends of the DNA, and then it will try to tether the it will try to attach the DNA to ends of DNA. Okay, and this will actually this process is actually a error prone process. Okay, so every time it tries to patch up the two ends of DNA by this non-homologous end joining, it will try to add some nucleotides, or it will try to remove some nucleotides. Okay, it will try to add some nucleotides or remove some nucleotides from the joining ends. Okay, so you can see like uh, um, from the cut end, some nucleotides can get added during the joining process or ligation process, and some nucleotides sometimes get removed from the cut site. So after patch up, after it's ligated, you will see either insertions or deletions at that original cut site. Okay, so after ligation, you will see this one. So actually, this non-homologous end joining is carried out by several proteins. Okay, out of which the human DNA ligase 4, that plays a very important role. In fact, if you add any inhibitor of this enzyme, this non-homologous end joining will not take place. Okay, so this is one of the predominant mechanisms of repair in the human cells. Okay, the non-homologous end joining mechanism. So it recruits several proteins, clue proteins, and many other proteins. So nearly some 50 proteins have been identified which can bind and then try to patch it up. So out of which the human DNA ligase 4 is the most important one, and because that is the one which eventually ligates the cut ends. But this ligation is a quick and error-prone process, leading to insertion deletions at the cut site. And these insertion deletions, what will happen? What will happen? If the insertion deletions are in the coding sequence, it will lead to frame shift, right? The reading frame shift will happen, and this reading frame shift will introduce premature stop codons. Okay, so as a result of premature stop codons, you'll end up having non-functional or like truncated versions of your protein, which will generally be non-functional. Okay, so this mechanism of repair will eventually lead to a gene knockout. Gene knockout. Okay, so it ends up patching the DNA, but eventually the gene is knocked out. Okay, so this mechanism can be used for gene knockouts. So the thing is, you have to select a gene which you do not want. Okay, so suppose you want to study uh, what is the effect of this gene. So you knock out the gene and then you study how the cell behaves. Okay, so you
Yeah.
use CRISPR there. So what you do is you you just take the sequence of the gene, you identify a unique 20 base pair stretch within the gene, which is unique to the gene, like it's not present in other genes. Okay, basically you want to minimize off-target editing. You want only that gene to be targeted, not other genes. So you end up selecting using bioinformatic tools. You end up selecting a unique stretch of DNA within the gene, and then you design the guide RNA, and the guide RNA will go and bind there. It will cleave. Okay, the Cas9 guide RNA complex will cleave that sequence. That will cause a double-stranded break. So now the cell will try to replace this double-stranded break, and it will recruit these proteins. Ligation will happen, but it will be error-prone, and this will lead to premature stop codons formed, and that will lead to a non-functional protein. Okay, and that means that gene is knocked out. Okay, so this is how people make gene knockouts. Now on the other way, many cases like for example sickle cell disease and many other diseases where the precise location of the mutation is known, and you want to do some editing, or in many cases you want to insert some gene, some missing gene is there, you want to insert that one, or you want to repair some gene precisely. In those cases, non-homologous end joining is not useful because it will always lead to knockout. Okay, so you cannot repair a gene. So that's why there is another known mechanism called homology directed repair, HDR.
Mhm.
So this mechanism is active, but it's not very active. So, for example, 80% of the time, non-homologous end joining is the most active. 20% is HDR. But when you try to put some inhibitors, like inhibitor of human DNA ligase 4, like SCR7, so you add this compound SCR7, it will inhibit this human DNA ligase 4, then the non-homologous end joining can be suppressed. So then homologous direct repair will get a chance to repair. Okay. So now if we see in homologous homology directed repair, you add a third component that is called as the repair template. So if you take the normal CRISPR, you have Cas9 and you have guide RNA, right? You have Cas9 and you have guide RNA. These are the two elements. But when you want to do a specific or precise repair, or when you want to do a gene knock-in of a gene, then you need to have a repair template. Okay, you need to have a repair template. The repair template, if you see, it will have the region to be inserted and it will be flanked by homologous arms. Okay, can you see this one? Either side it is flanked by homologous arms. These homologous arms actually match the sequence of the region where the cut is made. Okay, so because of this homologous arms, you will end up having exchange of those fragments. That means whatever repair template you are giving, okay, you are the repair template is like how how Cas9 and guide RNA are sent inside the cell by transfection. Similarly, repair templates are also given. Okay, the repair templates can be a single stranded DNA, or it could be a double stranded linear DNA, or even it could be a plasmid. Like you can take the repair template and you can circularize it and make it a plasmid. Okay, so it is available in many forms. So this repair template will always have a gene or a sequence to be inserted or a sequence to be modified, and it will have the homologous arms, which actually are similar in sequence to the cut end of the DNA. Suppose you are cutting the sequence here. This is the sequence you're cutting. Okay. Now, what you do is this 1000 base pairs from here, 1000 base pairs from here. This sequence you graft. Okay. So this is the supposed region of cut. So either side, 1000 base per 1000 base pairs you take, and then you put your gene of interest in between. This gene will be inserted, knocked in. Okay. So these are the homology arms. So this sequence actually is copied from the area where you cut. Okay. So that is copied here, the sequence. Now this gene, now this is how you make a repair template. The repair template, when you send it inside, it will actually swap with this cut DNA and the gene of insert will go here. So the repair template, what you give as a gene, that will get knocked in and it will enter the genomic DNA. It will become part of the genomic DNA. Okay. So that is what is shown here. So this is actually a powerful way to knock in genes and also to precisely edit existing genes. For example, in sickle cell disease, I told right GAA, which codes for glutamic acid, that has become GTA. Now you want to revert that one, you have to use such things. Okay, so you can make a repair template. You can either way you can make 1000 base per homology arms, and instead of GAA, you can make instead of GTA, which is there in the faulty gene, you can put the correct sequence GAA, and then you can put 1000 base per 1000 base per homology arms. Okay, and this will become the repair template. So when you add Cas9 guide RNA and the repair template, then successful editing can happen, which will repair the gene. Okay, so this is one theoretically it should work. Okay, understood?
Yeah.
But the only thing is the probability of this happening is low. So we need to come with some techniques to improve this repair process. Repair through HDR, homogeneous repair.
Hmm.
Now we'll see how one can design the guide RNA, because all of the specificity and all of the efficiency of this cleavage and the repair, eventual repair, that depends upon how well we design the guide RNA, right? So if the design of the guide RNA is wrong, you end up making unwanted edits at other genes, leading to some complications. So, for example, in gene therapy, people use very specific Cas9s, which and they use bioinformatic tools for designing the guide RNA. So let us now go into the process of designing guide RNA. Okay, so yeah. So in the guide RNA design, the important steps are, first you have to identify a PAM sequence. Okay. So first you have to identify, the first important thing is what gene I want to target. So you first get the sequence of the gene. Okay. And in the sequence of the gene, you need to identify a unique stretch. So how do you identify the unique stretch?
With the PAM?
You
The
need to
PAM.
use bioinformatic tools. You need to use bioinformatic tools to identify the unique
Oh,
stretch. Okay. Okay, because like that region that you try to target, that should not be present in other genes, or even similar sequences should not be present in other genes. Because if similar sequences are found elsewhere, your guide RNA Cas9 complex will go and cleave there also. And therefore, you will end up editing at multiple unwanted places, leading to more complications. Okay. So the first thing is actually to identify a PAM sequence. Then 20 base pairs ahead of the PAM you have to select. And that sequence you have to copy. Can you see? This G, right? This GAACCC, this up to this. Okay. So I'm selecting this sequence. Now I have to just take this sequence and graft it before the scaffold sequence of my guide RNA. Right? So the scaffold sequence of the guide RNA will not change, right? So the scaffold sequence of the guide RNA will remain the same. I need to just take this sequence without the PAM. Without the PAM, only this sequence before the PAM I will take and graft it here. So this is how I design a guide RNA, right? So guide
mhm.
RNA spacer sequence is taken from the region which it wants to target. And the remaining is scaffold sequence, which is unique for the particular type of Cas9, right? So this is how people make this very simple. But only thing is, how do you know that this particular stretch that you selected is unique to this gene? Right? It may be present in other genes also, very similar sequences. Maybe one or two nucleotides may be different, but still, it may be similar. So the guide RNA that you design can go and bind there also. Right? For example, out of 20, if 17 or 18 are matching, then that also can happen, right? You can go and bind there also. So that is why we need to make sure, that uh, uh, the sequence that you select for cleavage, that is very unique to that gene. Okay. Now, next to what you do, suppose your PAM could be in the either strands. It could be the other strand also, the reverse strand also. Like this is the 5' to 3' strand, the other strand, like the reverse strand or the minus strand we call na. So minus strand, you can have the PAM in the minus strand also. But in that, since the 5' starts from the right hand to left hand, so you can see that the PAM is NGG. It is read this way, NGG. Okay, and you should take sequences from here to here, and then you have to just kind of take this, put 3', 5' here, and just invert that. Okay, just reverse that. So that is what and this sequence should be integrated as a it will be like grafted as the spacer sequence uh, along the scaffold to form the guide RNA, right? So this is how, one actually designs guide RNA. Okay, but it is simple. But since, you need to really assure that that sequence is unique, you need to use a bioinformatic tool rather than do it manually. Okay, so I will expose you to a bioinformatic tool. Have you heard about Chop Chop?
Chop chop.
Yeah.
No.
Okay, so I will tell about this Chop Chop. Okay,
Uh-huh.
so I'm able to see my screen.
Um, let's see. Yes, I
Yeah.
am.
So this Chop Chop is actually a web server based software tool, which is developed by Harvard researchers, Harvard University
Oh,
researchers. Yeah. So this is quite a very popular tool for designing the guide RNA, particularly for selecting the unique target sequences. Okay. So your aim is to knock out a gene. Okay. So for example, I will tell you there is a gene. Okay, if you go to gene cards, you're able to see my screen, right? There is a gene.
yeah.
Okay, that gene is called NRF2. It's also
Yeah.
called as NFE2L2 transcription
Okay.
factor. Okay, so the thing is, uh, like um, this actually NRF2 is actually involved in some inflammation and other response to injury and all those things. Okay, uh, so the thing is, if I want to knock out this gene, okay, I want to knock out this gene NRF2. So I need to just take its full name. This is the gene name, right? NFE2L2. Okay, so this is the gene NFE2, like BZIP transcription factor 2. Okay, so this is a gene. It has some role in inflammation and some response for injury. And so I want to knock out and see what happens. Okay, now how do we design a knockout system?
Hmm.
Now, well, like what you do is go here, type the target gene. So you just paste the target sequence, gene name. Okay, so you have the option to paste the target, or you can just paste the or you can just type the gene name. Okay, there's no need to put the entire sequence. So you just put the gene name. Okay, so NFE2L2. So what I want to do, I want to knock out the gene. So I select knockout. Okay, so this is the human gene. So I'm selecting Homo sapiens because it's a human gene. Okay, I'm selecting the latest assembly. Okay, the HG38 is the latest assembly of the human genome, and I want to use CRISPR-Cas9. So the normal CRISPR-Cas9, not the variants. Okay. Now,
Yeah.
how do I go? So if you go here, you have several options,
Options.
right?
Okay.
Yeah.
Yeah.
So since I'm going to use SP-Cas9, I'm selecting NGG PAM,
Okay.
right? And if so suppose I'm using some other Cas9, I'll be selecting the appropriate PAM sequences. Okay.
Okay.
That means like for known uh, every known Cas9, there exists a PAM. Okay.
Mhm.
So if you see this one, okay, so yeah, this is the another tool. If you can see here, can you see this one? For
Yes.
SP-Cas9 from Streptococcus 5 genes, this is the PAM sequence, 5 prime NGG 3 prime, right?
Yeah.
So
Mm-hmm.
for example, if you take this one, okay, AES-CPF1 from Acidomonococcus, okay, or from Lacanospiraceae. So you can see that the PAM sequence changes, right? It becomes TTTTN, right? So depending on the type of Cas9 you use, the PAM sequence will change. Okay. So suppose I'm using a conventional Cas9, SP-Cas9, then it's NGG. Okay. So that's why I'm selecting here. Since I'm so we are planning to use NGG. Okay. Now uh, the spacer sequence length, the guide RNA spacer sequence length should be around 20 base pairs, right? So we put 20 base pairs. Okay. So it should be around 20 to 22 only, not less than 18, not less than 22, not more than 22. That is ideal. 20 is ideal length for the
Okay.
target sequence of the guide RNA. Now, what we do, we as I told, we have to look for any similar sequences. Okay, I am trying to select some sequence within the gene, within the NEF2L2 gene. Okay, but are there sequences similar to the gene to the gene elsewhere? Okay, so I have to select a sequence within the gene which is unique and which is not found elsewhere in other part of the genome. Okay, so how do we do that? So I just try to find the off targets, possible off targets, given up to 3 mismatches. So my protospacer region, what I'm selecting, okay. So is there any sequence which is similar? It may not be identical. Even similarity is enough for off target editing. You understand? Now I'm talking about off target editing. Off target means other than the intended target, we are the cast and go and edit the unwanted off targets. Okay. So to minimize that one, we have to select that particular sequence, target sequence, which is unique to the gene. Okay. So up to 3 mismatches, I'm trying to find the off target. Okay. Because beyond 3 mismatches, it is no more similar, right? For example, if you have a 20 base pair stretch of protospacer, okay, and you have, for example, I'm having 20 base pair stretch of protospacer. Okay. I'm having another area, another region, somewhere, for example, this is in chromosome 1, suppose. Okay. Chromosome 3. This is in chromosome 11, suppose. Suppose there exists some gene, some sequence. For example, here, my 20 base pair stretch, and this 20 base pair stretch in the chromosome 11. So they're very similar, except that there are, here it was A and here it was G, 1 base pair difference. Okay. So we say that this off target has a mismatch of 1. Only 1 mismatch is there. Okay. So that means this is the potential off target. Okay. And even if there are 2 mismatches, for example, instead of A, you have G here. Instead of C, you may have T here. Okay. So we say that there are 2 mismatches. Okay. And this can exist in chromosome 12 or chromosome 14, whatever. Okay. So we say that you have one off target that is in chromosome 12, which actually happens because of 2 mismatches. And then we can go for 3 mismatches. So it was T here and it may be G here. Okay. And we say that, okay, there exists a similar sequence in somewhere, maybe in chromosome 19 somewhere. And that happens to have 3 mismatches with our protospacer. So the problem is the guide RNA that you design based on this, right? I showed now how we are designing guide RNA. So we are designing based on the protospacer, right? So the guide RNA that is designed to bind to this sequence, we can also bind to this sequence also, right? Because only 3 base pairs are different, na? And the remaining are all same, right? So it can bind here also and it can cleave here also. So instead of cleaving the gene that we want, the on target, we end up cleaving off targets also. Okay, which should not happen. So that is why we need to make sure that the region that we select does not contain similar sequences in other places. Okay, so that is why we use the bioinformatic tools. The other thing is beyond 3 mismatches, they're quite for example, beyond 3 mismatches, suppose 4, 5, 6, 7 there. Then what happens? Your guide RNA will not annihil there because beyond 3, it can recognize as something which is very dissimilar, and therefore it will not cleave. Okay, but within 3 mismatches, they look very, very, like only some out of 20, only 3 are actually different, right? So therefore, it can confuse and go and bind there also and cleave there also. So that is why we need to minimize off target effects, and that is why we are using bioinformatic tools. Now, so these are the options for this software. Now, what we do is we go and set this. So Cas9 is there. We select PAM. We select 3 mismatches. We calculate we calculate the efficiency score of on-target cleavage, because like being a mammalian gene, okay, these mammalian genes, they are all wrapped up in histones, yes or no? You know about the euchromatin, heterochromatin structure of the chromosome? Hello?
Oh, yeah, sorry. I didn't know I was on.
Yeah.
Yeah, I
So
do.
the chromosome, some regions are tightly packed, some regions are loosely packed,
Yeah.
right? We call this the euchromatin, heterochromatin, all those things, right? Yeah.
Hmm.
So that means the DNA, particularly the human DNA or the mammalian gene DNA, mammalian DNA, it's not same throughout. Some regions are highly accessible. Some regions are highly, uh, packed with the histones and they're not accessible. Okay. So, and some undergo modification also, like methylation, all those things. Okay. So that means like, the efficiency of cleavage at a particular site will depend on the sequence and whether it's bound to the histones, whether it is modified, all those things. So some scientists have come up with some algorithms, because the PAM should be accessible. If the PAM sequence NGG is actually bound to the histones and not accessible, then what will happen? It will not be able to bind the Cas9 protein, right? So the accessibility of the PAM sequence is very important, and people and that will actually make sure the cleavage happens or not. So people have developed some algorithms which can scan through and find which region of the DNA is available, which gene is buried, and all those things. And that is the efficiency score here. And also the complementarity check, self-complementarity check. That guide RNA contains this spacer sequence followed by the scaffold. Suppose the spacer sequence that you design based on the target sequence, if the spacer sequence happens to actually have some complementarity with the remaining part of the guide RNA, that is scaffold part of the guide RNA, then what will happen? The spacer will end up binding to itself, to the guide RNA itself, right? Instead of being available to bind to the target DNA. So we have to make sure that self-complementarity within the guide RNA does not exist. The spacer sequence, particularly spacer sequence, should be free. It should not actually anneal with itself with the scaffold sequence of the guide RNA. So that's what we do a check, self-complementary check. These are different options, sir. Okay. So what we do, we go and find targets. Now it's queued up. Maybe in 1 or 2 minutes, it'll show the results.
Okay. Do I just hit submit?
Yeah.
All right.
Can you please repeat?
Yeah, I'm trying. Wait, I don't see the submit button.
You're not seeing?
Uh, no, I like just put all the requirements in, but now I don't see the submit button.
You're not seeing the screen now.
Oh no, I see yours. I see yours.
You're seeing the screen, right?
Yeah, I can see yours.
How I set up you saw? How I set up you saw?
Yeah, I see your screen.
Yeah. So when I ran, this is what I get. Okay, it's quite fast. The server is quite fast. Okay, so it has identified some target sequences within the gene. Okay, that means like we can use that sequence to make the guide RNA. So these are target sequences. So we use some several target sequences, like for example 131 target sequences. Okay, because there are several PAM. Wherever there's a PAM, 20 base pairs before it will be taken and can be considered as a potential target sequence. But it'll start ranking all these target sequences. So which one will be the ideal one for us? So the top ranked ones are the ideal one for us. So how it is given the top rank for this? If you see here, all of them end with the PAM sequence. Can you see? G, G, G,
Yeah.
G, right? So everywhere G, G. So it ends with the PAM sequence. And the first 20, so it's totally 23 bases will be given here. Out of which the 20 bases, they constitute the 20 bases. The first 20 bases is the target sequence. And the remaining 3, so the first 20 bases is the protospacer, the remaining 3 is the PAM. Okay. So this is found in chromosome 2. So this NFEL2 gene is found in chromosome 2, and this is the location, genomic location. And we are targeting the plus strand. So the GC content of this target sequence looks good. Generally, the GC content should be like 30 to 70% or 30 to 80%. So 40 is actually good score for GC content. And the self-complementarity is 0. That is very good, because we don't want the target sequence or the guide RNA sequence to actually bind to some other sequence. Okay, so the target sequence, the self-complementarity is 0. That means when I take this target sequence and graft it as a guide RNA spacer sequence, that should not have complementarity with the remaining portion of the guide RNA, the scaffold portion of the guide RNA. So that's what we are checking, and that is the self-complementarity is 0. That's good. Now we are considering different, for example, for these 20 bases, I'm just giving a mismatch of like for mismatch of 0. That means exact sequence, whether it's repeated elsewhere, no. So the off target is 0. For mismatch of 0, MM0, off target is 0. So for mismatch of 1, suppose out of this 20 bases in the protospacer, any one out of the 20, if it's changed to some other any other nucleotide, whether it will look similar, whether it will be there in some other chromosome or some other place, right? So the thing is, that is also 0. The off target is 0. That means you don't have any off targets given a mismatch of one base. Okay. Suppose given a mismatch of 2, like we are just looking for similar sequences which are just defined by only 2 bases from the protospacer that we have selected. So that is also doesn't exist in the genome, in the human genome. So it shows 0. The off target for a mismatch of 2 also 0. Now off target for mismatch of 3, that is also 0. Okay. So beyond 3 mismatches, you really don't worry, because it will be dissimilar. So your guide RNA will not bind there. Okay. So up to 3 mismatches we checked there, and the off targets are all 0. And the efficiency of cleavage also is good. That means that region is quite accessible also. That region is quite accessible. Okay. So the efficiency of cleavage also is good, 79.5. So that means by using this target sequence to make a guide RNA, you'll end up having a guide RNA which is highly efficient on the on-target sequence. At the same time, it will show no off-target activity. It will not cleave unwanted regions. It will cleave only the gene of interest. Okay. So that is what is actually the thing. So if you see the ones which are lowly ranked, okay, you can see the self-complementarity may be there, and it may have multiple off-target possibilities. Okay, can you see for the mismatch of 2, there's 1 off target, for mismatch of 3, there's 7 off targets.
Yeah.
The efficiency of cleavage is low. That is why they're lowly ranked. So we have to select the highly ranked sequences, okay, the rank 1 or rank 2, like that. And then we have to take the first 10 bases, first 20 bases. First 20 bases you have to take, because the remaining one are the PAM, right? So if you go here, it shows rank 1, and it shows can you see? The last 3 are the PAM, and the remaining, the one shown here are the 20 bases. The first 20 bases are the one which we need for making the guide RNA, right? So we select this first 20 bases, and then we add the scaffold sequence of the guide RNA and to form the complete guide RNA, right? So if you see here, can you see this one? The top. So you can see, you can expand and see. You can see that all these guide RNAs are actually against the coding sequence only, right? The one shown in blue color is the coding sequence, the exons. Okay, so this is exon, this is also exon, this is exon, this is exon. And this is also exon. And in between them, you have very long intron. Can you see? The red line is the long intron. Introns can be quite long, and the exons are short sequences. So exons are the coding sequences. Because for a knockout, you have to cleave within the exons only. Okay, then only the reading frame change will happen after repair, and that will lead to knockout. If you end up cleaving somewhere here, okay, somewhere here, if you cleave in the intron sequences, that will not have any consequence, because when the cell tries to repair it, there will be indel mutations. But since it's a non-coding region, indel mutations in non-coding region will not cause any kind of effect. Okay, so that means like if you want to do a knockout, you have to target the coding sequence only. Okay, a gene may have coding as well as non-coding, right? It will have introns as well as exons. So that means you have to select the unique sequence within the exons, within the coding sequence, coding region of the gene. You got my point, huh?
Yeah.
Yeah. And preferably the initial exons, not the last exons. Okay. Because, like if you target the initial exons, from there onwards, the reading frame change will happen, and that may lead to, a premature stop codon elsewhere. Okay. So the protein will get truncated there itself. But if you end up editing the last exon, the significant portion of the protein will be formed, and only the tail region of the protein, the C termini of the protein, that may have some change. Okay. And so the protein will be still functional. So that means you are not achieved a knockout. So in order to do a knockout, particularly for genes with multiple exons, you need to target the initial exons, either exon 1 or exon 2, instead of the last exons. For example, like this gene has 5 exons. Okay, we need to target the initial exons. So
Mhm.
this is how we actually end up doing. Okay, understood? So this is
Yeah.
what is for a gene knockout. Suppose I want to do a gene knock-in. Okay. So what I do? So suppose I want to introduce a gene. Okay. For example, have you heard about P53?
About what? PF.
P53 gene.
No.
We call it TP also, T53. This is actually tumor suppressor protein. Okay.
Oh, yeah.
It's a kind of master regulator. It plays a very important role in suppressing tumors. Okay. Now, this gene is there in our body, but sometimes mutation in this gene can render it inactive. And therefore, those patients who have mutations in this gene, they can have, they can develop cancer. Okay, they can develop onco problems. So the thing is, what about supplementing this gene? What if we can knock on extra copy of this gene in case the gene is inactivated? So the thing is, like we can always take any gene and we can do a knock-in. But where can we do knock-in? Because you have chromosomes, right? And which chromosome you will do knock-in? And by knocking in, whether any existing gene will not get perturbed. So these are very important questions, right? So generally, for doing a knock-in of a gene, what we do, we have to select the sequence. Okay, either a non-essential gene can be taken, and that we can cleave, and then we can do a knock-in of this p53 or whatever gene we want to knock in. Okay, but there are some safe harbor sites. Safe harbor sites means like those regions which actually you can introduce a new gene without perturbing the, or without affecting the stability of the chromosomes. Okay, or without perturbing other genes. So such a thing is important for a knock-in. You cannot generally randomly knock in anywhere, right? Because some other vital gene may be disturbed because of this knock-in of a gene, or it may end up destabilizing the chromosome structure. So that's why we need to be careful when you do knock-in of a gene. So generally, there are some safe harbor sites. For example, have you heard about adeno-associated virus?
No.
So adeno-associated virus is used in generally in gene therapy, and this adeno-associated virus, it has safe harbor sites. That means when it tries to integrate the gene of interest into the human genome, it will actually end up at certain locations. Okay, and these locations are generally safe, which will not perturb the other sequences. Okay, so these are called AAV, adeno-associated virus safe harbor sites. So you can actually identify those sites and you can knock in your gene of interest there. As I said, if you want to do a knock-in of your gene of interest, you have to make sure that the HDR repair process is active, homologous repair active. So how will you make it active? By designing a repair template. So Cas9 guide RNA against the guide RNA should be targeted against the gene which you want to cleave. Okay, and the repair template should have the gene which you want to insert, uh, and it should be flanked with the homology arms. Okay, homology arms which will be identical in sequence to the uh, region where you want to cleave. Okay,
Mhm.
so for example, I'm taking a protein. Have you heard about the twin babies who have been edited by Chinese
Oh yeah,
scientists?
yeah, in China.
Yeah. So you know the story. There, that particular doctor, who was eventually, faced some legal proceedings. So
Yeah.
he actually managed to do editing of the human babies. Okay. So
Mhm.
how we achieved this, because the parents of those babies had HIV. Okay.
They had what?
So he HIV,
What is that?
AIDS disease, AIDS, human
Oh,
immunodeficiency.
yeah, HIV. Okay, okay.
Yeah. So the parents of those babies had HIV. So the thing is, if they want to conceive, the baby also will get HIV, right?
Yeah.
So to prevent that one, what they did in vitro fertilization. Okay. So they took the cells from the from the from the parents, and then
Yeah.
they did in vitro fertilization,
Right.
and they did the CRISPR editing, and they targeted the CCR5 gene. Okay. So why they
Mhm.
target the CCR5 gene is CCR5 gene is actually is actually non-essential gene. Okay, it does not have any any important function. Okay, so its function is like even without the CCR5 gene, people are healthy. Okay, so what is the significance of the gene? People is not it understood. Okay, but the thing is, if patients who actually have deletion of this gene or mutation of this gene, CCR5 gene, they are resistant to HIV. So that is a positive trait, na? By deleting this gene or by knocking out this gene, okay. So when you do a knockout of this gene, the the babies can have HIV resistance, right? So that means like even when they are in the that HIV mom's womb, they don't develop HIV. Okay, they don't develop AIDS because the CCR5 gene plays an important role. The CCR5 gene codes for CCR5 receptor, which actually helps in internalizing the HIV virus. Okay, by knocking off this gene, you can make those cells resistant to HIV. So that is what he did. He knocked off that CCR5 gene, and therefore those babies, though they are born for HIV patients, they ended up being healthy without they didn't acquire the disease. So that was actually success. But actually, since he tried it without getting permission, he was eventually
Arrested.
arrested. Yeah. But actually, he proved an important point that, like one can play with the genes and one can prevent diseases. Right.
Yeah.
So he targeted CCR5 gene. So what we do is, suppose I want, so what we can do is two mangoes in a single shot. We can remove an unwanted gene. I mean, we will not remove, we can knock out an unwanted gene or a perturb an unwanted gene. And in that place, we can introduce a
Second
gene
one?
which we want to supplement. A gene which we want to supplement. So
Okay.
I want P53 gene because this gene plays a beneficial role. And CCR5 gene, it plays a negative role, right? It only helps HIV to enter inside. So I want to knock out CCR5, and there I want to introduce the P53 gene. So that means like I have to select CCR5. This is the target sequence, target gene, because I want to cleave this gene, right? And then I select knock-in because I want to cleave this gene and then do a knock-in. Okay. So I want to identify sites. Can you see?
Yeah.
It is there in chromosome 3, and this is the target sequence. And you can see that GC connect is good for target sequence. The self complementarity is good, and the mismatches up to two mismatches, the off target is 0. For mismatch of 3, there's one off target. But the thing is, if you go here, if the mismatches are close to the PAM sequence, this is the PAM, na? TG is the PAM, right? So in chromosome 11, this gene is in chromosome 3, but the off target is in chromosome 11. Can you see it's shown in chromosome 11? Okay. So
Yes.
when you account for 3 mismatches, like the exact sequence is not found, but but a similar sequence, not exact, but similar, just with 3 mismatches difference. The mismatches are shown in red here. But since this mismatches are close to the PAM sequence, though this is a possible off target, most probably it will not cleave here, because the mismatches seem to be in the seed region of the of the protospacer. Seed region means very near to the PAM. So whenever the mismatches are near to the PAM, that will not end up cleaving. Okay, so this chromosome 11 will not, this sequence will not get cleaved. But if the mismatches, whatever shown in red color, the mismatches, if it happens to be away from the PAM, maybe like the initial sequences here, okay, somewhere here, then cleavage will occur. Then we have to worry. But in this case, we need not worry, because though it is recognized in off target, but the mismatches happen to be near the PAM sequence. Therefore, the off target editing will not happen. Okay. So we can say that, even for this 3 mismatch, off target editing will not happen. Okay. So that means we can use this sequence. You can use it. So you just take this sequence up to here, then the remaining scaffold sequence of the guide RNA, you patch it up, you form the guide RNA. Okay. And you complex the guide RNA with the Cas9, and then you nucleofect the cells. So these will go inside, and then they will do the cleavage. All right. So the thing is, that is not enough, because you need to knock in the gene that you want to knock in, that you're not considered right now. So that will be in the repair template. Okay. So the target gene is CCR5, because you want to knock out the gene, and you want to introduce T53 gene. So T53 is there in the repair template. So you do knock in, then you develop a repair template, which will have the P53 gene. And then it will have the homology arms. These homology arms will be around 1000 base pairs, 1000 to 1200 base pairs. And how the homology arms are designed? So in the CCR5 gene, whatever CCR5 gene that you're trying to target, so if this is the cleavage site, if this is the cleavage site in CCR5 gene, okay, so this guide RNA will go and bind and cleave here, CCR5 gene. So from this cleavage point, 1000 base pairs here, 1000 base pairs here. These sequences only are taken and grafted here in making the repair template. Okay, you understood?
Yeah.
And this is the P53 gene will come, and it will occupy here in the cleavage site. So in the finally, you'll end up having something like this in the genomic DNA. P53 gene will get integrated into the genomic DNA. Okay. So this cell will have a functional copy of P53 gene. So just I showed P53. You can introduce any other gene you want. Okay. So for example, I can take a gene present in a plant pathway and introduce it into yeast, and I can make that yeast produce some useful compound, which is produced only in plants. This kind of synthetic biology is now possible with CRISPR-Cas9. In fact, in our laboratory also we are doing such things. We are trying to make yeast cells produce certain compounds which only plant cells can produce. Plants can produce. Okay. So that biosynthetic machinery can be actually taken, and those genes can be knocked inside yeast, and yeast can produce those things which can, for example, sandalwood. Have you heard about sandalwood?
Mm, like the
Sandalwood,
scent.
sandalwood. It's like having good aroma, right? Aroma.
Yeah.
Yeah, and even they add hop plants in beer. That all the whatever that gene which is responsible for the aroma and flavor, those genes present in the plant, they can be actually knocked in inside the yeast genome, and the yeast will produce those flavors. Okay, so along with the beer, it will produce a flavored beer. You don't need to add any kind of plant tissues inside. Okay,
Hmm.
so this kind of synthetic biology is possible. In fact, many kind of bacteria have been edited. Okay, so that they produce valuable metabolites, which only plants can produce. Okay, so whatever the biosynthetic machinery which is exists in other organisms, you can take it and graft it inside and you can create an engineered strain, a newly engineered strains, which have a lot of industrial applications. So CRISPR has got a lot of industrial applications, whether it's gene therapy, like sickle cell disease, Duchenne muscular dystrophy, cystic fibrosis. The list goes on, right? And a lot of developments have happened. And even in T cell therapy, have you heard about CAR T cell therapy? Chimeric
Sorry.
antigen, chimeric antigen receptors T cell therapy, where we
Oh
take the
wait.
patient's T cell.
Oh
We can
wait.
take the patient's
Sorry. Go ahead.
T cell therapy, T cell.
Yeah. I heard.
Yeah. So we can take the patient T cells and then we can do CRISPR editing of the T cells. We can change the T cell, we can do the editing of the T cells, gene editing of T
Yeah.
cells. And those T cells, we can knock in a synthetic receptor, which will identify some cancer cells. Okay. So
Yep.
you actually do a knock-in of a receptor which can identify tumor antigens. Okay. So that is actually grafted into the T cell genome. So the T cell will be able to now, these edited T cells will be able to identify cancer cells and destroy them. So those T cells which are edited are injected back into the body. So those will go after the cancer cells and kill the cancer cells. So this
Yeah.
is called as chimeric antigen receptor T cell therapy. So this is also like making a lot of news, and this is another important achievement of CRISPR, that you're able to make you know uh, cancer therapy possible. Okay, so
Mhm.
now the other thing is you have you have designed guide RNA. You'll be you know design how to design repair template, right? You know how to design a repair template. You know how to design a guide RNA. Now the other thing is how do you validate the edits? How do you know? You have to do an experiment, na? Like
Yeah.
you are adding all these components inside by nucleofiction, by transfection. They go inside. They cleave. But what about confirming them whether the really that area has got edited? How do you confirm?
I don't know. AI.
You have to do sequencing, DNA sequencing. Okay,
Alright.
so you have to sequence before the edit, and you have to do sequencing after the edit, after the CRISPR editing. Okay, and then these that will show the difference. So the before the edit, you'll have the normal. I mean the that whatever is there in the patient. Okay, after the edit, things would have changed, right? For example, sickle cell disease, before the edit, that GAA, GTA will be there. After the editing, it will be GAA, right? So you need to confirm that. So you need to do the sequencing and see, right? So you have to do DNA sequencing. There are two types of DNA sequencing. One is the Sanger sequencing, and which actually we take small region of the DNA sequence, only the region of interest. And in other type of sequencing is the NGS. That is the next generation sequencing. Have you heard about next generation sequencing?
I think.
Next
Um.
generation sequencing, NGS.
And it sounds familiar. Uh.
Yeah. So in
Is
next generation sequencing, the advantage is you can sequence the entire genome. The
yeah.
entire genome, human genome, can be sequenced and analyzed within 1 or 2 days. So the advantage is not only the on-target editing, but also any unintentional side effects like off-target editing. That also can be assessed by NGS. next generation sequencing, because you are thinking that this will target your gene of interest. But what if it is perturbed? Though you are using bioinformatic tools to minimize those off targets, sometimes it can happen, right? So you need to always sequence the entire genome and see whether any unwanted cleavage elsewhere has happened. Okay, and if it's not so, then it's good. The treatment is successful. Okay, so that is how one has to validate, and there are some software tools for that. For example, have you from one? There's one thing called Tide, right? Just once again,
Right.
Tide, TAD, Tide Decision.
Oh.
This is from a London-based company, which are able to see this series.
Yeah.
Tide. So this is actually a software tool to assess the editing
Sequence?
performance. Whether after editing, you need to confirm by sequencing. Okay, so the sequencing reads will be taken and subjected to bioinformatic analysis using this tool called TIDE, which will tell whether a knockout has happened or not. Okay, so it's a rapid and easy assessment of genome editing, because you need to know now, like just by having a raw sequencing file, you'll not know now. So this can take up the file and read that file, and it can tell whether editing has happened or not. Okay, so if you go to this tide, tide. So basically here I'll just load the example here. Okay, I will just load the example data. So basically you have to give the sequence. Okay, you have to give the guide sequence. Whatever guide RNA you use, that guide sequence you have to give. You have to give the chromatogram of the control sample. That means before you started editing. Now, for example, you have a bunch of cells in your tissue plate, your cell culture plate, okay, which you want to edit. So that will be the control. Okay, so before editing, so you will have a control cells. Control means you don't edit those cells. So those the DNA from those cells are taken, and then you do PCR, and that PCR product is given for sequencing. Okay, and then the edited cells, where you have treated the CRISPR constructs, the edited cells are taken, and then you isolate genomic DNA, and you do PCR. You take the PCR insert and you PCR product, and then you give for sequencing. So this is the sample chromatogram. Whatever you generate is a sample chromatograms. Okay. So you understood now what is control, what is sample. Yeah.
Yeah.
The control is the wild type. That means like unedited cells, right? And the sample is the one which you have edited by CRISPR. Okay. So both these should be sequenced. Okay. So that chromatograms raw, like in this format, SCF format or AB1 format, raw chromatograms, you can directly upload to the software. Okay, and you can upload the guide sequence, and then you can actually get information about the editing. Can you see here? It says that
Yeah.
62.8% of cells, okay,
Yeah.
is actually having no edit, because it's zero. Neither insertion has happened nor deletion has happened. Right? That means 60%, more than 60, like 60% is not undergone any change. But you can see 23.2% has undergone deletion, and 5% has undergone a +1 insertion, one nucleotide insertion, and 2.1% has got +2 deletion. Two nucleotides got deleted, okay, at the cut site. So that means that 23, all these will lead to knockouts. That means 23.2 plus 5 plus 2.1. How much? 28.2 plus 2. It will be almost 30 percentage. Okay, almost 30 percentage is actually undergone knockout, because all these will produce knockout, right? This will produce knockout, right? Yes or no? Yeah, this will
Yeah.
produce knockout, because there's a one nucleotide deletion, -1. This will also cause knockout, because this is 2 point. So this is also will cause knockout. This is +1 addition. All these are indel mutations, right? These will all lead to knockout. But this one is no change. So this means unedited, no change. It will remain as such, like the wild type. Okay. So now you have a mixed pool of cells after editing. Somehow got edited, these ones, red ones, and this one, 60% one. This is not edited. So you have mixture of cells which are edited, and whatever chromatogram you get will have this signals from all these things, right? So you need to delineate and find out the respective frequencies. How much is unedited? How much is edited? And what are the different deletions and insertions? How much percentage of cells are undergone deletion? How much percentage of cells are undergone addition? All these things you have to validate, right? So that is why you need to see all these things, right? So that means you are delineating and finding out the respective frequencies from that chromatogram. If you can see, this is the cut site. Okay, up to here, up to the cut site, you can see everything is normal, right? The wild type one and the edited sequence all match here, right?
Mhm.
But after the cut point, you can see that there is a mix-up. Can you see? Right? The edited ones and the wild type ones, they are all not matching. Okay. So maybe like if you can see in the other view, it'll be there better. Okay. So if you see this one, can you see?
Mhm.
Like for better picture, this is the wild type one, and this is the mixed pool of cells. Up to here, it's normal. Can you see?
Mhm.
You can see that these two match up to here. Can you see this up to here? It's same, all same, right? The wild type and the mixed pool of cells are all same. This is the one before editing. This is the one after editing. Okay, this is the control. This is the sample. Sample has become mixed pool because some are edited, some are not edited, some are edited with the multiple deletions, some are edited with the insertions. So that's why it's called mixed pool. So up to the cut point, they're all same. But afterwards, you can see in the mixed pool, you have overlap. Can you see? There's overlap,
Yeah.
right? One signal overlapping with another. So you get actually a composite signal because you have many species now, like with some deletions, with some insertions, without any insertions or deletions. Like that, you have a mixed outcome. So we have to deconvolute. We have to decompose this complex signal into these three, right? So that is what the software does. It takes this composite signal, Sanger sequencing composite signal, and delineates into wild type one, or with the one with the insertions, or with the one deletions, and then gives the respective percentages. So this is wild type one is still 34%. The one with the deletions is 30.3. With the multiple deletions is 17.2. With the one single insertion is 14.1. So all these ones, 17.2, 30.3, 14.1, they will lead to knockout, right? So
Mhm.
we can say that in this case, it's like some 60% will need to knock out. So this is how one can actually validate the CRISPR editing.
Yeah.
Okay. So shall I stop here? Any questions?
Yes. Yes, please. Not really. I'll like, okay, I've understood how, like, what you showed me about the two websites, but like it's like
Yeah.
I'll probably
So
pretty
that chop
yeah.
chop from Harvard, and this Tide for validation, and chop for designing the guide RNA. Repair
Okay.
template design is very easy. Just you need to take the homology arms, and then you just based on the based on
Yeah.
the flanking ends, you just need to graft it around the gene of interest. Okay.
Mhm.
So
Yeah.
overall, you understood now, like what is CRISPR, what are its applications, and how we select the target sequence, and how we do the use the bioinformatic tools for selecting the target sequence, how the guide is designed, how the repair template is designed, and you know how we do the editing, and how we validate the edits. right? How we validate edits. So all these things we saw. Okay. So this is the most essential part. We also offer 5-day courses also. This is just a 2-hour course, a 5-day program, like a 2 hours daily for 5 days. Okay. That will have, for example, I offer CRISPR-based gene therapy course, which is actually having some 12-hour session. Like it'll be like split into 2.5 hours daily for 5 days. Okay. So that will give you a more complete picture of this CRISPR based gene therapy.
Okay.
Okay. So thanks for your patient listening. If you have any
Yeah,
questions, you can ask.
um, let's see. Let me just review my notes.
Recorded videos will be given to you. Okay. So you can watch the recorded videos,
Oh, okay.
and you can email to me also. Okay.
Yeah.
Any
Right.
doubts you have, right? So I'll
Yeah.
just share my email ID. Uh, So this is my email ID. In case you have any doubts or you are looking for some any kind of uh, you know uh, help related to these topics, then you can contact me.
All right.
Okay.
Thank you.
Okay. Thanks. Bye.
Thank you. Bye.